Welcome to the lecture on the development of the nervous system. The nervous system starts developing extremely early. You may recall from anatomy that fertilization of the egg takes place not in the uterus typically, but in the uterine tube. While still in the uterine tube and prior to implantation in the uterus, the egg has started to divide, and it has now become a sphere of cells or a group of cells. And within these cells, you get a cavity formed, and this occurs before the embryo actually implants in the uterus, so very, very early on. This sphere of cells then develops into three layers: an ectoderm, a mesoderm, and an endoderm. The ectoderm is going to form the nervous system, as well as our sensory organs and the epidermis, so the outer layer of skin. The mesoderm is going to form the dermis, the area of skin underneath the superficial layer of cells, our muscles, our bones, connective tissues throughout the body, and the circulatory and urinary systems. And the endoderm is going to form our respiratory and GI systems. Specific development of the nervous system begins at approximately day 18 of gestation. On one side of the sphere of cells that now has a cavity in it, a group of cells start to line up with each other. This area where they're lining up is going to eventually be the dorsum of our embryo, so it's the back. This group of cells that is lined up together is called the neural plate. The neural plate then begins to fold in on itself. It begins to buckle, similar to if you took a piece of paper and held it on each end and then brought your hands closer together. The middle of the paper would fold in on itself, making a little valley. On either side of that little valley, there are what looks like two small hills. Those hills are called the neural folds, and the valley is called the neural groove. The neural folds continue to grow and enlarge, and eventually they come into contact with each other. Our neural groove has now been completely closed off, and it is now called the neural tube. This has only taken about 3 days from start to finish. At the same time that the neural tube is formed, cells that are off to the side of our neural folds are pinched off or separate away from the neural folds and neural tube. And these particular cells are called the neural crest cells. The formation of this neural tube is why our nervous system in mature individuals is hollow. So here is a picture that depicts what's going on. So we are on what will be the dorsum of the embryo. We have this neural plate, or this group of cells that have lined up with each other. And then we have off to the side, you can see there are a group of cells that are going to eventually become the outer layer of our skin, the epidermis. The neural plate begins to fold in on itself, creating a valley and then two small kind of hills on either side. So the two small hills are represented in green, and those are the neural fold, with the valley being the neural groove. As those neural folds continue to grow, they come in contact with each other, which closes off the neural tube. And we have cells then that are pinched off on either side, and those are the neural crest cells. You'll notice that the neural tube and neural folds, which now create the upper part of the neural tube, also separate themselves off from the cells that will form the epidermis, or outer layer of skin. Our neural tube is going to develop into our brain, our spinal cord, all of our motor efferents, so this would be both voluntary motor neurons and preganglionic autonomic motor neurons, and our glial cells, those cells we talked about in the prior lecture that are so important for providing protection and scaffolding to our neurons. The center of the neural tube itself will become the central canal of the spinal cord and the ventricles of the brain. The neural crest cells, those cells that were pinched off to the sides, will become our sympathetic trunk and the postganglionic autonomic neurons. They'll also form part of the adrenal gland and pancreas. And they'll form our dorsal root ganglion and somatosensory neurons. So the neural crest cells essentially are going to be forming the peripheral nervous system, that is where the cell bodies are located outside of the central nervous system, whereas the neural tube is going to create the central nervous system itself, plus those peripheral nerves who have their cell bodies inside of the central nervous system. So again, our preganglionic autonomics and our voluntary motor, their cell bodies are located in the spinal cord or brainstem itself, with the axons then traveling peripherally to get to the muscles that they're going to innervate. Whereas our somatosensory neurons, they have their cell bodies outside of the central nervous system in a ganglion, as well as our postganglionic neurons for our autonomics. They're also located outside the central nervous system. So those are formed by the neural crest cells. This closure of the neural tube progresses in a specific order. It starts in the cervical region and then progresses superiorly and then progresses inferiorly. So the first place for the neural groove to close and become the neural tube is in your neck. And then that closure, the enlargement of the neural fold, large enough to actually close off the groove and make it a tube, happens superiorly before it's completed inferiorly. Now they're not like it happens superiorly and then when it's done inferiorly starts. It's just that superiorly is completed prior to completion of an inferior closure. If there is a problem with closure of the neural groove in either direction, then you will have some form of pathology. If it fails to close in the superior direction, you have what's called anencephaly, or lack of a brain. If it happens inferiorly, you have a condition known as spina bifida. So where that happens can determine the extent of the defects. Now, next to the neural groove and neural tube, we have mesoderm. And the mesoderm that is next to this these neural structures divides itself into cell clusters. They're kind of spherical shaped, but they're probably a little more egg-shaped than truly round. But that doesn't really matter. What I want you to know is that the mesoderm groups itself into these little clusters we call somites. The somites are divided into 3 different areas. I'm not going to ask you to know which specific part of the somite is going to be what structure, but what I want you to know is that what a sclerotome, a myotome, and a dermatome are, and that they come from the somites. So one part of the somite is going to be the sclerotome. These are the cells that are going to form our vertebra and ribs and part of the skull. Another part of the somite is going to be the myotome. This is going to form our skeletal muscles. And the third part of the somite are the cells that are going to form the dermis, the remainder of our skin. And this is called the dermatome. And this is why we have segmentation in the nervous system and why we have certain muscles that are formed from certain segments and certain areas of skin that are formed and innervated by certain segments. Here you can see a picture of an embryo. Its neural groove has begun to close, but you can see it's still open at the top and it's still open at the bottom. So we don't have a complete neural tube yet at this point. We still have those neural folds looking like kind of little hills or ridges on either side, and the neural groove in the center. But on either side of that, you can see these kind of roundish clusters. Those are the somites. And so again, those somites really are like having three groups of cells all clumped together. One group of cells going to become our sclerotome and form our vertebra and ribs and skull. One group of those cells is going to form our muscles, and one group of those cells is going to form the dermal areas of the skin. And so each of those somites is going to come off of a particular part of the brainstem or spinal cord. As the neural tube has completed closure, we'll find that the cranial end of the tube starts to bulge and become larger. This is going to become the forebrain, the midbrain, and the hindbrain of our nervous system. The forebrain is then going to significantly grow. It just is expansive, and it divides into two parts. So the forebrain is made up of our cerebral hemispheres, and you'll recall we have a right and a left cerebral hemisphere. So that is what happens after our division. It also includes our basal ganglia, and it includes the diencephalon. And you'll recall the diencephalon is made up of the thalamus, hypothalamus, subthalamus, and epithalamus. The midbrain, as we said earlier, is the mesencephalon. And then the hindbrain is made up of the rest of the brainstem, the pons and medulla oblongata, as well as the cerebellum. This is just a picture to show you where these things are relative to each other. So we're looking at a mid-sagittal view. This the brain and brainstem has been cut right down the middle, like through your nose, and you can see the cerebral hemisphere, so the forebrain up at the top, and then in green and orange are two parts of the diencephalon, the thalamus and the hypothalamus. Inferior to that, you have the mesencephalon, the midbrain, and then it's not labeled on here, but inferior to that, where kind of that egg-shaped object is, that's the pons, and then below that is the medulla oblongata. Now, specifically, what is happening with the cells as this brain is forming? Well, first, cells are undergoing mitosis, so they're dividing, making more cells. And we have stem cells, and there are stem cells for different tissues, but we have neural stem cells that prior to birth can become either neurons or they're supporting glial cells. Cells then migrate to the correct location of where they need to go. And they do so by following a glial cell that has a very long tail. How this glial cell knows where to go is not known yet. We don't really understand how they know where to go, but these glial cells with these kind of long tails will make their way along the nervous system to get to a particular location, and then the neurons that form will follow this tail, kind of like climbing a rope. The neural cells then differentiate or specialize. This appears to be determined not by a predetermination within the cell, but instead by the location where the cell ends up migrating to. For instance, they've taken, in some studies in animals, they've taken some neural cells that were migrating into one area and they've moved them to another area. And their specialization became correct for the new area. It was not based on the area that they were originally heading to. So it does seem at this point that while we have specialized functions of different nerve cells within the brain, that the location may determine it and its connections to other cells may determine its function, as opposed to having it predetermined when it's first formed from that stem cell. Myelination begins to take place at approximately 20 weeks of gestation. Myelination takes a long time. While the majority of myelination is done by the age of about 3, We still have areas that continue to require myelination all the way up into, in some cases, the early 20s. This is particularly true for young men. Women, for whatever reason, females do myelinate more quickly than men. And the last area to fully myelinate in both sexes, both men and women, is the frontal cortex. And the frontal cortex, we will learn, is responsible for such things as impulse control, understanding and thinking about consequences of actions, organization, planning, all of these kinds of what we call executive functions. So you'll know that teenagers tend to be rather impulsive and have a harder time keeping some of these things under control. And they're not always all that well organized. Part of the reason is because their frontal cortex has not completely myelinated yet. The other thing is that when it comes to potty training, boys typically are slower to potty train than girls. And this is likely due to the fact that they're slower to myelinate the pathways that go from the cortex or from the cerebrum into the brainstem and spinal cord to help control urination and defecation. So because it takes longer before the boys have full myelination, that's probably at least in part why boys tend to potty train later than girls. At the time of birth, you have twice as many neurons as you will have as an adult. So right now, you have half as many neurons as you were born with. Your peripheral nerves were well myelinated when you were born, especially those for sensation. The cranial nerves involved with tasting and sucking and swallowing were also well myelinated. This makes sense, it's for survival. And higher order neurons for the sense of smell are myelinated. Again, that makes sense because for survival, the infant must be able to locate the food source. However, the rest of the central nervous system is quite poorly myelinated. The brain does a lot of growing early on, and it is going to double in size within the first year. During childhood, you continue to myelinate those CNS neurons that were not myelinated at the time of birth. Now remember that their peripheral neurons are pretty well myelinated already at birth. Synaptogenesis is taking place, so we are forming connections between neurons. We're forming communication connections. So they don't physically come into contact with each other and hook together. There's always a slight gap, a few micrometers in between. But this is where the neurons communicate to each other. So we're forming synapses. Neurons that fail to create a synapse or have a very inactive synapse are going to likely be removed. The neuron will die, and the synapse that's there will be taken away, and this process is called pruning. There is a critical period or a sensitive period for learning different things, and these periods occur at different ages. But during these critical periods, this seems to be a time when synaptogenesis is particularly maximized. The ability to form these new synapses seems to be enhanced at certain periods of time. And these synapses tend to lead then to essentially permanent connections. They're very hard to eliminate. This is one of the reasons that things like learning a language, a second language, is easier when you're young than when you're older. Because when you're older, you're past this critical or sensitive period where you've maximized your ability to create synapses. And the other thing is axons are going to compete for synapses. So those axons that, again, are not as strong, you're not getting as much activity there, will be pruned away. As teenagers, myelination of the central nervous system is continuing. We're particularly working in the areas of the prefrontal cortex. Again, remember that's planning and goal setting, follow-up, /U is follow-up. So recognizing that you need to reflect on things and make adjustments for them. The corpus callosum, which is a connection between the right and the left hemispheres of the cerebrum, is getting myelinated. The emotional centers are starting to myelinate more so. So you find that young teenagers tend to be much more emotional than and emotionally labile, where they just seem to cry and then suddenly they're laughing and then they're back to crying. And then when you get into the later teen years, like 18, 19, there seems to be a little bit more stability there. And the higher order language centers are also myelinating. We're continuing to form new synapses, but we're also continuing to prune out neurons that are not making connections or synapses that are not being active. And now I want to talk about a few pathologies of neural development. I do want to first, though, make sure that you understand I do not want you to memorize pathologies. This is true throughout the entire semester. There will occasionally be some pathologies that I will ask you to know. And I'm going to ask you to know terms related to particular dysfunctions, such as what it means if I say ataxia, or what it means if I say agnosia, or other terms of that nature. Okay. But when it comes to a specific diagnosis, as opposed to a sign or symptom, unless I'm telling you otherwise, you do not need to memorize those particular conditions. I'm giving them to you just to help you to see from a clinical standpoint how some of these damage or impaired functioning within a particular process impacts individuals on a higher level. So for the pathologies or the conditions that I'm going to discuss, again, if I were to put them on an exam, I would tell you what they are. But I would pose a question that would make sure that you understand the underlying process that went awry. The first developmental disorder I want to talk about is anencephaly. This is where the neural tube fails to close on its superior end. No forebrain, as a result, develops. So this infant is born without the cerebral hemispheres, the basal ganglia, or the diencephalon. And typically the skull is incompletely formed. These infants will frequently be stillborn, or they'll die very shortly after birth, within the first few days. There are cases, however, of infants living for a longer period of time. And just recently I read about a young boy who lived actually for several years. Now, to give you an example of how I might use this information on an exam, I would not ask a question that were to say, anencephaly is, and then have you fill in the blank. But I might ask a question such as, anencephaly is failure of the neural tube to close superiorly. Prior to the failure of closure, closure of the tube was occurring normally. Which part of the neural tube was completely closed prior to the anencephaly developing? Cervical, thoracic, lumbar, or sacral? What this would require is that you understand that the neural tube closes first in the cervical level and then it progresses superiorly before completing inferiorly. So in order to answer that question, you would go, well, anencephaly is the failure of it to close superior to the cervical region, but that doesn't start happening until the cervical region is closed. So cervical would be the correct answer. So again, you don't have to know what anencephaly is, but you need to understand the process of closure of the neural tube and the general order that things take place in. The next disorder is spina bifida. I mentioned spina bifida is failure of the tube to close inferiorly. There are several different kinds of spina bifida. everything from simply missing the vertebral arch, so the pedicles and laminae did not form correctly on one particular vertebra, leaving a gap. This is typically asymptomatic, although the person may have a tuft of hair over that area where the missing vertebral arch is. I'm not sure why that tuft of hair grows, but it seems it's a common occurrence. Well, many times this is discovered while taking an x-ray for something else that's totally unrelated, and the individual has all normal function. But it can also go to the opposite extreme, where you have complete protrusion of the spinal cord and meninges out of the spinal column defect, and that is accompanied by severe neurological impairments. Spina bifida also appears to be probably a syndrome, because many times there are other symptoms, not just related to the spinal cord, that are seen with individuals with spina bifida, such as cognitive issues. Spina bifida has been associated with folic acid deficiency during pregnancy. So women who are pregnant or planning to get pregnant are given folic acid supplements. Paralysis and sensory impairment will occur of varying degrees for all individuals who have involvement of the spinal cord and meninges. As I mentioned, for those where they are simply lacking the vertebral arch, but the spinal cord is perfectly in place, the meninges are good, nothing else has been affected, they will be asymptomatic. But others will have varying degrees of paralysis and sensory impairment. This video is just a quick video from the Spina Bifida Association. Just kind of talks about spina bifida. It's very short, very overall generalization, but it kind of gives you a feel or a sense for spina bifida. Arnold-Chiari malformation is where the cerebellum is, sometimes part of the brainstem, but basically we're talking the cerebellum, is dropped down through the foramen magnum, the opening in the bottom of your skull where your spinal cord goes through. And it compresses onto the inferior brainstem and the upper spinal cord. There are a couple of different types of Arnold-Chiari. Type 1 is where the individual has a small pons and medulla oblongata. Because they are smaller than normal, the cerebellum is able to protrude down through the foramen magnum into the vertebral canal. This results in compression of the cerebellum. It may result in hydrocephalus, which is a blockage of cerebral spinal fluid. It's almost always associated with headaches in the suboccipital region, and it can be associated with cerebellar signs. And when we talk about the cerebellum in Unit 3, you'll have a good sense of what those different symptoms, signs and symptoms could be. Type 2 Arnold-Chiari, the medulla as well as the cerebellum protrudes into the vertebral canal. This always shows up in infancy. Type 1 may not show up in infancy. It may show up later. I know somebody who just this past year had surgery for type 1 Arnold-Chiari for a malformation, and she's in her early 20s. But type 2 pretty much shows up in infancy. Hydrocephalus is majority of the time, I'm going to say. I'm not going to give you a percentage because I wouldn't know off the top of my head. But the majority of cases, these infants will have hydrocephalus. And they often will have a meningomyelocele, which is a type of spina bifida. So you've got this abnormally small pons medulla. Your medulla and your cerebellum are down within the vertebral canal. They're coming out of the skull and protruding into the vertebral canal. And you've got a defect in the inferior aspect of the vertebral column and the central nervous system. So you've got problems really from both ends with neural closure and development of the nervous system. Our next developmental disorder is Down syndrome, or trisomy 21. This is a very common disorder. The incidence of Down syndrome appears to increase as mothers become older. So women are putting off childbearing now for careers, and then having children at later ages. And the incidence of Down syndrome does appear to be related to the mother's age, with a higher risk of having a child with Down syndrome for older mothers than for younger. Some of the developmental issues related to Down syndrome are a smaller brain overall with fewer neurons, fewer dendrites than normal, so there's not as many places for synapses to form, so you don't have as many connections between neurons. A marked decrease in synaptogenesis. So again, if you're not creating new synapses, you're not going to be able to learn as well, because learning, as we will find out, is the creation of new synapses. The cerebellum appears to be larger than normal, but many of the cells within the cerebellum are not differentiated. So having a larger cerebellum doesn't mean that it works better, because the cells within it aren't really differentiated to do specific jobs. Individuals with Down syndrome often will develop an Alzheimer-like syndrome. We're not sure if it's truly Alzheimer or if it's something similar. I've seen kind of mixed opinions in the literature. And for reasons unknown, individuals with Down syndrome don't get cancer as much as the general population, with the exception of leukemia. They do seem to have a higher risk for developing leukemia. Fetal alcohol syndrome, the ingestion of alcohol during pregnancy, and usually with binge drinking. So binge drinking has been found to be more problematic in the formation of fetal alcohol syndrome than drinking on a daily basis. So even if the overall amount of alcohol intake is the same at the end of the week, if it was done in large doses on a binge, it seems to have greater impact than if it's spread out over that week. But obviously, we want to minimize alcohol intake during pregnancy at all times. But fetal alcohol syndrome interferes with gene expression for the development of the central nervous system and ends up with widespread malformations as a result. These children will be hypotonic, so they'll be very floppy, ragdoll-like, have poor coordination, and cognition will be decreased. They'll have difficulty learning, and cognitive impairments will show up. There tend to be behavioral problems and growth deficiencies. Additionally, there are some facial anomalies that are characteristic for infants with fetal alcohol syndrome, just as there are certain facial anomalies that are consistent for individuals with Down syndrome. The shape of the eyes or the distance of the eyes from each other, and a few other features that are somewhat consistent. So it seems to be particular genes that are affected more so than others by both of these two conditions. Epilepsy is a form of seizure disorder. There are several different categories of epilepsy. Causes include genetics, birth trauma, traumatic brain injury, that's TBI, or tumors. Under genetics, you can have heterotopia, and that means that neurons that are in the wrong location, they're not where they're supposed to be, and this seems to be due to failure to migrate or to migrate incorrectly and creating abnormal action potentials. So again, this is a little mixed from what I said previously, that some of the, when it comes to differentiation, some evidence seems to indicate it's not where you end up, or excuse me, it's where you end up, not what if you were meant to be a certain thing when you started. Heterotopia, we kind of see signs that it is dependent right from the get-go when the stem cell becomes a neuron as to what you're supposed to do. But it may be more not the differentiation as much as the activity levels, that certain neurons maybe from the start are meant to be more active than others. We also see genetic abnormalities in ion channels, and we'll talk about ion channels next week, for chloride and potassium in particular. But with epilepsy, we tend to get uncontrolled and excessive brain activity or action potentials. And too much action leads to cell death. As I mentioned, there are different types of epilepsy. I'm just going to mention two different presentations. There's what we call generalized seizures and partial seizures. And the main difference is if it is multiple areas where seizures initiate, or whether there is one particular area that consistently is the starting point of these seizures. If someone has a head trauma or has a tumor that causes irritation and excessive excitability of the localized neurons, you're going to have partial seizures. You can have partial seizures without either of those, but you're unlikely to have a generalized seizure if it's because of a specific injury or a tumor in a particular area. And as I mentioned, too much output from cells is neurotoxic and will create death of neurons. And that's the end of our developmental lecture.